This is a special episode of the Immunology podcast, Immunology2025, Day 4. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rao. Welcome back to the Immunology podcast, where we have conversations with immunologists. Today we're back with our fourth episode from Immunology2025. Thank you to everyone who visited our booth and joined us to record today. Our on-the-ground episode featuring conversations with delegates will air on May 20th. As always, we'll be discussing our favorite sessions throughout the past 24 hours of the meeting, so if you weren't able to attend, we've got you covered. We're going to kick off things in just a moment, but before we get to that...
Do you work with human immune cells? Learn how to isolate highly pure immune cells and efficiently process human samples with a free on-demand course on human immune cell isolation. To learn more and register, visit stemcell.com/human immune course. All one word.
You work with human immune cells, don't you, Brenda?
I do. Yeah. So maybe I should get the course, maybe.
Do you ever, when you're desperate for samples, draw your own blood?
We're not allowed.
Really?
Yeah.
Like that's the most allowed thing in the U.S.
I know.
So there's all these rules to get other people's samples, but if you're like tap my own vein, go for it.
I know. I wish I had done that in my grad lab. We could have done that. I don't like needles. I never volunteered. But in my career, and I work in the blood bank of the Netherlands, right? And they're very careful with the whole privacy laws, and also the idea is that you don't wanna, if you're gonna like do something with these cells, genetically modify them or whatever. If you get contaminated with your own cells, your body cannot reject and something like that. So they, out of biosafety and just privacy, you're not allowed to.
Interesting.
I know.
We totally had to do some controls for our infectious screening, and there weren't enough donor samples, so various employees all donated to get, like, you need like 20 different people, right? That you do all spikes in. So we always lined up and people started donating. Yeah, no, I never volunteered for that. As I said, I don't like getting blood taken. It makes me crazy, and so I'm a bit of a wuss in that. So anyway, all right.
That being said, Day 4.
Wow.
We had our live show yesterday. We had our recordings today with the delegates. It's been a whirlwind of action here.
My voice is a little tired, but we'll make it work.
Yeah,
I'm on my
see that.
fourth thing of coffee already.
I know. Somebody asked you, "Are you okay?" You look very tired. One of our guests asked you.
And what you told them I was just old, so thank you.
Okay. You know what? Let's not waste too much time. You have a lot of science to go over.
All right. So, tissue-resident immune cells, homeostasis, and repair. My first one this morning. Boom.
There you go.
All right. So it's kind of the mucosa because you're repairing it, but it's not. It's more immune cells, but still mucosa because it's magical. All right. First one, the role of Treg expressed MMP-12 during the resolution of acute lung injury by Priya Bose from my alma mater, UNC Chapel Hill. Tar Heels. All right. So they find that MMP-12, a major metalloprotease, is upregulated in Tregs in acute lung injury models in mice. And when they knock it out, there's an inflammatory state with more immune cell infiltration. And so they do some kinetic work, look at proteins expressed. And the big conclusion they're able to find is that while MMP-12 is needed early on for inflammation, that's how immune cells chew through the matrix and get in where they need to get going. Later on, it's an anti-inflammatory, specifically in Tregs. But they don't know why yet. It's a science talk where sometimes there's mysteries of the universe to unsolve.
Yeah, open-ended.
Next up, from the University of Miami School of Medicine, with Sujad Yunis, a distinctive skin gamma delta T cell repertoire in diabetic wound repair. Key takeaways from this is the diabetic foot ulcers, which is a complication of people with diabetes where they get tissue injury that's poorly heals, have lower amounts of gamma delta T cells, and the quantity is dysregulated in diabetic skin. And they find that while commensal bacteria induce in these cells perforin 2, and it's kind of a big deal. The gamma delta T cells are producing perforin 2, which is antimicrobial peptide, but that's kind of that gamma delta role, right? It's kind of an innate cell, kind of sometimes, but not really, but a little bit. In this case, a little perforin. But, the high sugar environment in the footbed and everything else going on there leads to dysbiosis. It's always about the microbiome. And so commensal bacteria induce perforin 2, but pathogenic or pathobiont bacteria suppress its production. And so it actually suppresses the ability of the wound to kill other bacteria and then heal. Then we had connexin 43 mediated interaction between alveolar macrophages and type 2 epithelial progenitors protects against bleomycin-induced acute lung injury from Ganling Q of University of Florida. So alveolar macrophages are an important part of tissue repair in the lung. And there's a subset of them they identified that adhere to the epithelium of the lung by a connexin 43. And if you knock it out, you have worse lung injury in their models. And if you give them, and then if you do transfers, like here's some macrophages from a normal mouse, they can rescue it, alveolar macrophages. But if you use ones without the connexin gene, and you knock it out, the receptor on the other side, it also breaks. So you have to, for tissue repair of the lung, you need these alveolar macrophages connecting to the epithelium through connexin 43. So that was cool. Next up, this is one of those, oh my God, like we talked about on our live show, everything connects to everything else. Mm-hmm. So macrophages regulate pituitary hormone regulation independent of hypothalamic cell signal, from Zena Maria Del Mundo from UC Irvine. So they have a really cool system where they can target specific type of macrophages and deplete them with an adenoviral vector, which they went over real quick, but I'm just going to assume they do it like it's magic, which is cool. But they found that 3 weeks after 50% depletion of pituitary macrophages using the system, and they show that it doesn't deplete elsewhere, that you have a decrease in pituitary hormones produced by the pituitary without any change in the hypothalamic axis to control it. So macrophages in the pituitary help control hormone release independent of the main driver, which is the hypothalamic axis. Oh. So your macrophages help determine your hormones.
Wow. Anything they don't do?
No. Macrophages being my second favorite immune cell. They're, of course, awesome. And then right after this, went to another talk, scrambled over. I got to listen to one of these great ones. Unfortunately, my regular life in the microbiome sciences also caught up with me this morning for a little bit. But I got to listen to Influence of the Microbiota on Mucosal Immune Response and listen to a great talk with Lehan Morgan from UAB called Microbiota Antigen-Specific Regulatory T Cells and Colitis Prevention and Treatment. So they have a special mouse that they use called the CBER1TG. It is TCR. It's a specific mouse with a TCR, a specific TCR that is engineered and designed to be responsive to a specific commensal bacterial flagellin, right? So there's this flagellin TCR for commensal bacteria. And if you go and take these immune cells in an adopted transfer model and put it into a regular mouse or a mouse without an immune system, and you give it to them, they develop spontaneous colitis, and it mirrors pretty well IBD in a lot of ways. It has that nice anti-commensal microbiome thing going on. So then they have an IL-12, IL-2 mutein that preferentially binds the CD25 Tregs. It doesn't bind to the other cells that respond to IL-2 because they have different subparts of the receptor, and those drive inflammation. But this mutein only binds the IL-2 receptor on CD25 positive Tregs of the CD25 receptor, and so it drives Treg activity but doesn't drive the other IL-2 immune pathways that are inflammatory. So it's an anti-inflammatory IL-2 mutein. And when you give it to these mice, you have a massive spike in IL-10 producing Tregs that prevent the CD4-driven colitis. So new drug, new Treg, new drug to the rescue, druggable Tregging without gene engineering.
Very nice, very interesting.
So that was my morning before we had several hours of recording those delegates today.
Very cool. Very cool. All right. So from my side, so I went to the session about the pathogenesis of autoimmune diseases, the relationship between microbiome, microbes, and the autoimmune disease, the development, and it was really cool. And it's cool because, well, the first speaker was Hisashi Arase from Osaka University, and they actually covered a very recently published paper that we covered in one of our roundups. I don't know how long ago, but so it was nice to see being presented by the authors. So basically, the talk was titled "Self and Neo Self-Discrimination by T Cells in Pathogenesis of Autoimmune Diseases," and it is really cool because it brings a very intriguing kind of approach at the autoimmunity, and this kind of the origin of autoimmunity and the kind of targets that autoimmune cells are recognizing. And basically, they show that so MHC class II molecules, they're presenting antigens preferentially to CD4 cells, and MHC class II has this property that they are this feature that they are two separate chains, no, and they have this groove in between where your peptide will fit. Usually, the peptide of MHC-II molecules is a few amino acids longer than the typical class 1 peptide that has only one chain that is more rigid in the space of what can fit. But MHC-II, they're a bit more flexible in what fits and can be kind of presented by this MHC. So basically, the gist of this talk is that if you interfere with the normal assembly of the MHC-II peptide complex, you can have weird things happening. So normally there is this chain called invariant chain that is part of the MHC-II complex, while the complex is being kind of generated in the, I think would be in the endosomal reticulum or in the Golgi. And before MHC molecule gets loaded with its corresponding peptide that it's supposed to be presenting, the time before that, it's been kind of occupied by this invariant chain. And in a way, it kind of keeps, this invariant chain is important for reasons that are, they make very clear in this publication. If the absence of the invariant chain, because you're knocked out or because you have a virus that can downregulate the expression of this invariant chain, results in the presentation, kind of the binding and presentation of food proteins that were never meant to be presented like that, and that are presumably not being presented in the thymus, and therefore there's no tolerance against them. And so if you have a situation in which you have mice that don't have this invariant chain, and they start presenting in the surface things like IgG chains or things that are not meant to be presented, and this can trigger autoimmunity. This can become a target for autoimmune cells, and they actually show that in the case of lupus, of SLE, it really seems to be a major target recognized as self-antigen. Is this neo self-antigens that are basically the consequence of downregulation of the invariant chain and the presentation of this weird, big antigen proteins on the surface instead of like regular, properly cut and processed peptides. They show that something like in mice, they have that the TCRs are reactive to these kind of neo-self-antigen, in lupus patients compared to healthy donors, can be up to 10% of the clonally expanded repertoire that is they see in this patient, which is really, really a lot. So like making one of the biggest kind of contributors to SLE, at least in this patient that have this recognition. And what's interesting is that our good friend, the Epstein-Barr virus, comes up as a risk factor, and apparently there are some of the viral proteins from EBV can induce this downregulation of this invariant chain, and that could potentially be mediating this increased neo-self-presentation, and therefore, this weird the triggering of this autoimmune response. So
EBV is the worst.
I know, right? We should all it's too late for us, but we should get everyone vaccinated. EBV just keeps popping up. All this autoimmune diseases is always EBV somewhere lurking in the background. It's terrible. So they also did some, AlphaFold modeling to show how this would work. So it's pretty convincing. I thought it was really, really interesting and very intriguing proposition for SLE and other autoimmune diseases. So then later was James Moon from Harvard. It was funny. He started with a disambiguation, because there's another James J. Moon that also works on something, more or less immunological. And apparently they keep getting mixed up, and they keep getting like invitations and things for the other guys. So it's like, this is me working at Harvard, and this is my colleague working, I think it was Northwestern or something. Anyway, I thought that was funny. So basically looking at epitope spreading from microbial antigens to autoantigens. And basically the idea is to try to understand, why is it that often certain infections will initiate some kind of autoimmune disease. And the example that he gives is Lyme disease. And there's this bacteria, Borrelia burgdorferi, that the infection with this bacteria, which is spread by the deer tick. So it's something in common where there's ticks and deers. I think the U.S. is fairly well known. Not so much
Problem
in,
where I live.
yeah, not so much in Europe yet,
You guys
but I think
have
it's
deers
coming.
in Europe?
Yeah, but I think not so much
much.
Lyme. I think it's coming though. So usually most people can get treated with antibiotics, but a small amount will develop chronic arthritis. That's a problem. So he went about trying to understand, which are the antigens from this Borrelia bacteria that are close enough to self-antigens that might be the reason why you have this molecular mimicry. So they looked at PBMCs from patients. He identified potential mimic antigens and trying to find really identify which of those are having reactivity in both Borrelia, so against Borrelia peptides and endogenous peptides. So he found some of those. He also looked on some patients with rheumatoid arthritis, and I think this is a difference. Is Staph aureus, is it for rheumatoid arthritis? So the same idea. So he was really looking into peptides that would elicit responses in patients, and both when you have the original microbial peptide and the putative human target that has been actually erroneously targeted in this autoimmune disease. Then we had more SLE from Neda Aziz from Harvard Medical School, looking at the impact of different isoforms. Apparently B cells that are associated with lupus, for example, this called double negative B cells. They don't express either IgG-D or CD27, and these are kind of associated. They're expanded in autoimmune disorders, particularly lupus, and they're associated with this pathogenic B cell responses. And basically, she showed that these cells have this regulated splicing going on, and then you have these different isoforms that are presented in this population of cells. So they found when they compare, B cells from healthy donors from lupus patients, and they say that this IRF5 came up as a lupus risk locus, and that this that presents indeed a differential splicing isoform usage. And this different isoform seems to end up kind of increasing the expression of this IR5. And this IR5 increases proliferation and inflammatory cytokine production by these B cells. So kind of connecting these concepts in the disease. So they studied one particular risk variant. And another talk that I really like was from Anam Shaikh from University of Kansas, from the group of friend of the podcast, Robin Orozco. Big shout out to her and her group.
Hey.
And so in their group, they're working on this enzyme called PEP or PTPN22 in the human homolog. And this, it's a cell that is, I think, it's canonically a phosphatase. It's only expressed on immune cells, and it has a few interacting partners. This is CSK, which is involved in TCR signaling, and TRAF3, which is involved in type 1 interferon signaling. And they're studying this particular point mutation that happens, and then this I'm not sure where they got it from, but so they studied this particular point mutation, PEP-R619W, and this point mutation prevents the interaction with these two CSK and TRAF3, and it turns out that this variant is associated with higher risk of autoimmunity. So the question is also, could it maybe be used for viral immunity? How does it relate with viral immunity, which often is the flip side of these autoimmune phenotypes? And they show that in mice, this PEP variant clears in the viral infection faster. They have these LCMV models. It has enhanced production of interferon gamma. And interestingly, only against specific LCMV epitopes. So they tried like the most commonly used dominant peptide, and they didn't see any difference. But when they use a less, I think it's a minor epitope that is not, I think it's not so strongly recognized, that one actually made a difference to have this particular variant or not. And what it shows that if I understood correctly, this variant seems to affect interferon signaling differently in CD8 cells or in dendritic cells. So they show that in CD8 cells, so this PEP variant, it will result in reduced PSTAT1 expression after interferon beta exposure. So it does have like reduced interferon stimulation, of like these interferon ISGs, interferon stimulated genes. And this is not because of changes of the interferon receptor, but it looks like there's more expression of a negative regulator of STAT1, which is SOX3, and that this seems to be kind of the mechanism by which this particular variant works. And this is mostly seen in dendritic cells and not so much in T cells. So kind of indicating that there's kind of a immune cell specific regulation in which PEP in mice, and so actually the thing, PEP in mice or LIP in humans encoded by the gene PTPN22 is involved, and I think that's where they left the story for now. But basically that you have this particular signaling pathway that's affected by this protein through SOX3. And that is, there's cell type specific effects, which it's interesting from a therapeutic approach, maybe for chronic infections, if you could kind of stimulate interferon production by these dendritic cells, for example. And so, those were my highlights from the morning. I thought it was really nice. And then, we had a lot of recordings, and here we are. It's been great.
It's been quite excellent. Getting more and more friends to the podcast, I've noticed.
I know. Isn't that wonderful? I love it.
I love it too.
It's precious.
All right. Well, that brings us to the end of our latest Immunology 2025 episode with one more to go. Don't forget to follow us on X at ImmunoPodcast or Blue Sky at ImmunologyPodcast.com to find out what we're up to at the meeting. Check back here tomorrow for our final episode. See you then.